Lecture 33, Nature and Nurture. So far in this course, we've focused our attention on the mental life of mature adult human beings. We've taken mind and personality as givens and asked how they work. How does the mind work? How does the mind mediate the individual's interactions with his or her social environment? Now we want to take up a new question, which is, where does mind and personality come from? In addressing questions of mental development, psychologists tend to take one of three general views: the ontogenetic, the phylogenetic, and the cultural. The ontogenetic view has to do with the development of the individual human being. From the ontogenetic point of view, we trace the development of mind and personality across the lifespan of the individual, from birth through infancy, childhood, adolescence, adulthood, right through to old age and death. Most developmental psychologists focus on infancy and childhood, though with our aging population there's an increasing interest in lifespan development, extending into old age. This is the domain of developmental psychology, a major subfield in psychology, which often divides itself up into psychologists who are interested in cognitive development and those who are interested in social and personality development. These lectures are going to focus on the ontological view of development, and especially on personality development. But I'll also have some things to say about trends in the study of cognitive development. While the ontogenetic view is concerned with development of mind across the lifespan of the individual, the phylogenetic point of view is concerned with development across evolutionary time. To some extent, the phylogenetic point of view was expressed by comparative psychology, which, as its name implies, is concerned with studying learning and other mental functions in different species. More recently, evolutionary psychology, an offshoot of sociobiology, traces how our current human mental functions evolved through natural selection and similar processes. From the point of view of modern evolutionary psychology, much of the way we currently think and behave was shaped by the early environment of evolutionary adaptedness, basically the African savanna and the Pleistocene era about 2 million years ago, where the ancestors of humans first emerged. And finally, there's a cultural view of development, which is concerned with the effects of various forms of social development on the development of the individual's mind and behavior. This is represented in its early form in what's known as anthropological psychology, which tries to describe what the so-called savage mind is like. This actually is an offshoot of 19th century European colonialism, which often sought to contrast between the so-called advanced minds of the European colonists and the so-called primitive minds of those they colonized. But more broadly, it's based on the idea that there was a development in modes of thought as our human ancestors moved from the Stone Age to the Bronze Age to the Iron Age, and even in historical time as human beings moved from the ancient period through the medieval period, the early modern period of the 15th through the 18th centuries, the modern period, and now the period of postmodern thought. There's also literature on the role that literacy plays in shaping how individuals think and how cognitive development is correlated with economic and political development. These days, there's quite a bit of interest in what's known as cultural psychology, which recognizes a great diversity of mental life across cultures, without the implications that one culture is necessarily more developed than another one. As I say, these lectures are going to focus on the ontogenetic view of development, which is the way developmental psychology is usually construed. Throughout its history, developmental psychology has been focused on the question of the role of nature and nurture in mental development. This dichotomy between nature and nurture was originally proposed by Sir Francis Galton, a cousin of Charles Darwin's, who was in turn inspired by a line from Shakespeare's Tempest. Is the newborn child a kind of blank slate which is written on by experience? An organism that acquires its knowledge and skills through direct experience and vicarious social learning? Or are some aspects of mental life innate, part of the child's genetic endowment, acquired through the course of human evolution. As Galton defined these terms, nature refers to all that a person brings with him or herself into the world, the individual's genetic and hormonal endowment, his or her physiological constitution and temperament, whereas nurture refers to every influence on the individual that occurs after his or her birth, the influence of the physical and social environment, what the individual gets from social learning, socialization, and all aspects of experiential history. You can see the relationship between Galton's dichotomy of nature and nurture and the dichotomy between nativism and empiricism that we discussed at the beginning of our lectures on sensation and perception. The empiricist thought that all knowledge was acquired through experience, while the nativist thought that some knowledge was innate. Galton is making very much the same kind of distinction. Of course, there are lots of different ways in which we can think about the relationship between nature and nurture. Galton seemed to frame the problem as one of opposition, in which some characteristic of mind and behavior was due to nature or nurture. But of course, we can also think of nature and nurture working together, either exerting independent influences on some aspect of mind and behavior, or somehow interacting with each other to generate mind and behavior. I bet you can guess where this lecture is going to end up. Well, why keep it a secret? A major purpose of these lectures on psychological development is to illustrate what I think of as a developmental corollary to the doctrine of interactionism. In interactionism, we saw that the person is a part of his or her own environment and shapes that environment at least as much as the environment shapes the person. Similarly, I want to stress that development isn't just something that happens to the individual, with the individual passively on the receiving end of nature and nurture. Rather, the developing child, even the infant fresh out of the womb, is actively engaged in the developmental process and actively working as an agent of his or her own development. But we're not there yet. Let's begin our discussion of the ontogenesis of mind and behavior at the beginning with the human genome. The human genetic endowment consists of 23 pairs of chromosomes, each chromosome containing a large number of genes. Every gene is located at a particular place on a specific chromosome. And since chromosomes come in pairs, so do genes. Corresponding pairs of genes contain information about some characteristic, such as eye color, skin pigmentation, and the like. Some traits are determined by single pairs of genes. Others appear to be determined by several genes acting together. Two of these chromosomes, the X and the Y chromosome, determine the individual's sex. If you're XX, you're genetically female. If you're XY, you're genetically male. Though it will be clear from a subsequent lecture that things are actually a little bit more complicated than that. Usually, we think about genes as having to do with an organism's physical characteristics, but it's entirely possible that there is some genetic influence on the organism's mental and behavioral characteristics as well. One of the most important technical advances in recent biological research has been the decoding of the human genome, determining the precise location of the 20,000 or so genes that make us as a species different from all other species. And along with this advance came gene mapping, which makes it possible to determine specific genes that put us at risk for various diseases, or disposes us to various personality traits. Here you see Craig Venter, whose private research group contributed greatly to the deciphering of the human genome. And you also see Venter's own genome. These are his chromosomes, along with the identification of specific genes that, as far as we can tell, dispose him to heart attacks, give him a preference for evening over morning, genes linked to tobacco addiction, novelty seeking, Alzheimer's disease, and the like. Now, the presence of these genes doesn't mean that Venter is predestined for a heart attack or Alzheimer's disease, any more than he was genetically predestined to be a surfer, which in fact he is. It's just that these genes appear to be more common in people who have these problems than in those who do not. They're risk factors, but not an irrevocable sentence to heart disease or dementia. The entire set of genes comprises the organism's genotype, or genetic blueprint. An organism's genotype represents its biological potential. This potential is actualized within a particular environmental context to produce the organism's phenotype. So one's genetic endowment interacts with various environmental factors to produce the phenotype, or what the organism actually looks like. Because of the role played by the environment, phenotypes are not necessarily equivalent to genotypes. For example, two individuals may have the same phenotype, but quite different genotypes. Similarly, it can happen that two individuals will have the same genotype, but different phenotypes. Of two brown-eyed individuals, one might have two dominant genes for brown eyes, while the other might have one dominant gene for brown eyes and one recessive gene for blue eyes. Different genotypes, same phenotype. Or two individuals may have the same dominant genes for dimples, but one might have his or her dimples removed by plastic surgery. Same genotype, different phenotype. So the environment matters. And we need to distinguish among three different kinds of environmental influence. The prenatal environment is the environment of the developing fetus in the womb during gestation. The perinatal environment is the environment present around the time of birth, labor, delivery, and immediately afterwards. And the postnatal environment is the environment present after birth and continually changing throughout the course of the individual's life cycle from birth to death. Many basic questions of nature and nurture, particularly concerning personality development, have been addressed by using the technique of behavior genetics. And perhaps the most popular method in behavior genetics, really the gold standard for behavior genetic research, is the twin study, which, as its name implies, compares two kinds of twins in terms of their similarity in personality. Monozygotic twins, or identical twins, are the product of an egg that has been fertilized by a single sperm, but which subsequently split into two embryos, thus yielding two individuals who are genetically identical. Dizygotic, or fraternal twins, occur when two different eggs are fertilized by two different sperm, thus yielding two individuals who have only about 50% of their genes in common. The basic idea behind the twin study method is that if a trait is inherited, even in part, monozygotic or identical twins should be more alike on that trait than dizygotic or fraternal twins, because monozygotic twins are identical genetically, and dizygotic twins only have about 50% of their genes in common. The usual technique in twin studies of personality is to administer some personality questionnaire, like the NEO Personality Inventory, to a large sample of monozygotic and dizygotic twins, thus obtaining scores representing each individual's standing on each personality trait measured by the questionnaire. Then we measure the similarity of the twins on each trait by means of the correlation coefficient. You'll remember that this is a statistic that summarizes the direction and strength of the relationship between two variables. For example, between extroversion in one twin and extroversion in the other. If a trait were wholly inherited, we'd expect to see a perfect correlation between identical twins, a correlation coefficient of 1.0. And we'd also expect to see a substantial but not perfect correlation of 0.50 for dizygotic twins. If a trait is wholly inherited, then genetically identical twins should be identical in personality. And again, if the trait is wholly inherited, you would expect some degree of similarity in personality between dizygotic twins as well, because they are genetically similar to at least some extent. For genetically unrelated individuals, we'd expect to see no correlation. If a trait is wholly inherited, and if there is no genetic overlap, then there should be no similarity in personality. An alternative measure of similarity is the concordance rate. That is simply the percentage of subjects that have some characteristic in common. We'll look at some concordance rates when we discuss behavior genetic studies of mental illness, but the logic is the same. If some characteristic is wholly inherited, we'd expect to see a concordance rate of 100% for monozygotic twins because they're genetically identical. If one has the trait, the other must have it too. And we'd expect to see a concordance rate of about 50% for dizygotic twins, and a concordance rate of 0 for unrelated individuals. Again, to the extent that a trait is inherited, we expect that monozygotic identical twins will be more similar to each other than our dizygotic fraternal twins, regardless of whether similarity is measured by the correlation coefficient or the concordance rate. Here's a quick fictional example. This data is made up. Imagine that we had a set of 10 twins, 20 individuals, and we administered a 60-point scale of extroversion and simply determined their scores. You can see just by inspection that there's a high degree of correspondence between the scores of one individual and the score of his or her twin. It's not perfect, but in general, individuals with low scores have twins with low scores. Individuals with high scores have twins with high scores. In fact, if you calculate the correlation is almost but not quite perfect, a correlation of 0.99. Now here's some real twin study data, taken from a study by John Lowland and his colleagues, in which monozygotic and dizygotic twins were administered a personality inventory that contained scales measuring the big 5 personality traits. Studies using other personality inventories have obtained similar kinds of findings. You can see that on each of the big 5 personality traits, monozygotic identical twins are more alike than dizygotic fraternal twins. Results like this provide prima facie evidence for a genetic contribution to individual differences in personality. The simple fact that monozygotic twins are more alike than dizygotic twins strongly suggests that there's a genetic component to these individual differences. But the correlations for monozygotic twins are far from the perfect 1.0 that we would expect if a trait were wholly inherited. So it's very clear that genes aren't the only forces that are determining individual differences in personality. But if you think about it, data of the sort that I've just shown you doesn't provide wholly convincing evidence that there is any genetic contribution to personality. Sure, monozygotic twins are genetically identical, but monozygotic twins also share an environment that is arguably, anyway, more alike than it is for dizygotic twins. After all, monozygotic twins are of the same sex, and they also resemble each other physically. So it wouldn't be surprising to discover that monozygotic twins are treated more similarly than dizygotic twins are. For this reason, the very best twin studies, like Lowland's, employ samples of dizygotic twins that are of the same sex. This reduces, at least to some degree, the discrepancy in environmental experience between monozygotic and dizygotic twins. But still, the problem remains. How exactly can we tease apart the genetic and environmental contributions to personality? The question is complicated still further by understanding that, in fact, environmental influences themselves are very complicated. There are 2 different kinds of environmental influences on personality: the shared environment and the non-shared environment. The shared environment, also known as between-family variance, includes all the factors that are shared by children raised in the same family, factors that differentiate them from children raised in other families. As a rule, children in the same family are raised by the same parents, share a single racial, ethnic, and cultural heritage, live in the same neighborhood, go to the same schools, attend the same church, synagogue, or mosque. The shared environment includes all these things, and more, that siblings within a family have in common. The non-shared environment, also known as within-family variance, includes all the factors that differentiate among children raised in the same family. Even within a family, children can differ in terms of such factors as gender, whether they're boys or girls, or birth order, whether they're the firstborn or later born. Children may have different interactions with their parents and develop different networks of friends and acquaintances beyond the family. Different children within a family are also distinguished by non-systematic factors, which include all the things that happen randomly to one child, but not to his or her brothers and sisters. These chance encounters can really make a difference in the individual's life. The non-shared environment is an umbrella term that refers to all the unique experiences that siblings can have. As it happens, the relative strength of both environmental components of personality, as well as the genetic component, can be estimated from the observed pattern of monozygotic and dizygotic twin correlations. Consider first the entire distribution of a trait within a population, from those individuals with the lowest scores on extroversion or neuroticism to those with the highest scores on these traits. This distribution is typically represented by a more or less normal distribution, the famous bell curve. The entire distribution of individual scores within a population is called the total variance of the trait in question. This total variance on some trait like extroversion can be further decomposed into genetic variance, that is, the amount of total variance that's attributable to individual differences in genotypes, and environmental variance, that is, the amount of the total variance that's attributable to individual differences in environments. In principle, then, the total variance on a trait is equal to the sum of the genetic variance and the environmental variance, with the understanding that environmental variance includes both variance attributable to the shared environment and variance attributable to the non-shared environment. And, in fact, we can calculate the components of population variance, the genetic variance and both types of environmental variance, from the data given to us by twin studies. And here's how we do it. The genetic variance can be estimated by the difference between the monozygotic and dizygotic correlations multiplied by 2. To estimate genetic variance, you double the difference between monozygotic and dizygotic twins. Don't worry about where the 2 comes from. That's a technical detail. But you can get a sense of it if you just consider that if a trait were wholly inherited, the monozygotic correlation would be 1.0. The dizygotic correlation would be 0.50, so the difference between them is 0.50. Double that difference, you get 1.0. All the population variance would be attributable to genetic variance. That's the idea. So what's left? Well, what's left is the environmental variance. Variance due to the non-shared environment is a function of the MZ correlation. Monozygotic twins share both genes and the shared environment, so any monozygotic correlation less than a perfect 1.0 must reflect the contribution of the non-shared environment. It's just logic. If monozygotic twins share both genes and the shared environment, which they do by definition, then to the extent that they do not perfectly resemble each other, that difference must be due to what they don't have in common, and that's the non-shared environment. So we estimate the non-shared environment by subtracting the monozygotic correlation from perfect 1.0. And once we've estimated the contributions of the genes and the non-shared environment, variance due to the shared environment is all that's left. So that can be estimated simply by subtracting the genetic variance and the non-shared environmental variance from 1. Now, just to get the idea down firmly, let's work through a couple of numerical examples. Here's the ideal case, at least from a genetic point of view. The MZ correlation is a perfect 1.0. The DZ correlation is 0.50. Double the difference between MZ and DZ, and you get an estimate of genetic variance at 1.0. All the population variance is attributable to genetic variance. Of course, there's nothing left over, but let's do it anyway. The non-shared environment is estimated by 1 minus the MZ correlation. That's 1 minus 1, or 0. So there's no contribution from the non-shared environment. and we can estimate the contribution of the shared environment by subtracting from 1 the genetic variance and the non-shared environmental variance. 1 minus 1 minus 0 leaves 0. No contribution from the shared environment either. This is exactly what we'd expect if a trait were wholly inherited. Now, let's cut the MZ correlation in half. Monozygotic twins correlate 0.50. Dizygotic twins correlate 0.40. Double the difference, and you get an estimate of genetic variance of 0.20. 20% of the population variance is attributable to genetic variance. To estimate the contribution of the non-shared environment, 1 minus the MZ correlation of 0.50. 50% of the population variation is attributable to the non-shared environment. What's left? 1 minus 0.20 minus 0.50 equals 0.30. 30% of the population variance is attributable to the shared environment. Here's another one. Monozygotic correlation of 0.80, dizygotic correlation of 0.70. Double the difference, so 20% of population variance is attributable to genetic variance. 1 minus the MZ correlation, 20% of the population variance is attributable to the non-shared environment. And what's left is 0.60. 60% of the population variance is attributable to the shared environment. And one more. Monozygotic twins, correlation of 0.5. Dizygotic twins, correlation of 0.25. Double the difference. 50% of the population variance is attributable to genes. 1 minus the MZ correlation, 50% of the population variance is attributable to the non-shared environment. What's left is nothing. None of the population variance is attributable to the shared environment. Now let's look at some real data. This is data derived from family studies of IQ, measured intelligence, including twins. The general pattern here is that as individuals increase in genetic relatedness, they also increase in their similarity of IQ. Monozygotic twins are more alike in IQ than dizygotic twins, even when those monozygotic twins have been reared apart. Again, this is a very common pattern in family studies of IQ. Aggregating results from a large number of studies of this sort, we can estimate that the correlation between monozygotic twins for IQ is about 0.86, and the correlation for dizygotic twins is about 0.60. So now let's plug those values into our formulas. Double the difference between monozygotic and dizygotic twins, we find that about 52% of the population variance in IQ appears to be attributable to genetic variance. Subtract the monozygotic correlation from 1. About 14% of the variance in IQ is attributable to the non-shared environment. And what's left? About 34% of the population variance in IQ appears to be attributable to the shared environment. So the genes play a role in determining IQ. But genes are not the sole determinants of IQ. The environment is also important, and of our two kinds of environment, the shared environment seems to be more important than the non-shared environment in determining individual differences in IQ. Here is similar data from a twin study of educational achievement, how much schooling people get. Again, monozygotic twins are very alike on this. They show a correlation of about 0.86. Dizygotic twins show a correlation of 0.66. Double the difference, we see that about 40% of population variance in educational achievement is attributable to genetic variance. This makes sense. If education has something to do with intelligence, and intelligence has something to do with the genes, then genes ought to play a role in educational achievement. 1 minus the MZ correlation indicates that the non-shared environment contributes only about 14% of population variance. But now what's left over, 0.46, the shared environment now turns out to be not just more important than the non-shared environment, but also, at least marginally, more important than genes. Some families send their kids to school, make their kids go to school, make their kids go to college, even if the kids aren't all that smart. So the shared environment is much more important as a determinant of educational outcome than it is as a determinant of IQ. Here are some findings from a behavior genetic study of adolescent and young adult behavior, particularly sexual behavior. The investigators measured what they call age of sexual debut, that is, age of first sexual intercourse. These investigators estimated that about 31% of variance in age of sexual debut was attributable to the genes. Only about 10% of the variance is attributable to the shared environment. The overwhelming proportion of variance, almost 60%, was attributable to the non-shared environment. And I'll leave it to you to speculate about precisely what those non-shared environmental factors might be. Here are the results from another study, this time from suicidal behavior. I showed you some results from this study in the introductory lecture. The investigators measured both suicidal ideation, the extent to which people thought about committing suicide, and also suicide attempts, whether they actually tried to do so, including completed and uncompleted attempts. It turns out that there's a genetic influence on both suicidal ideation and suicidal attempt, though the genetic influence is stronger for ideation than it is for attempt. The shared environment played apparently no role in suicidal ideation, and just a little role in suicidal attempt. But the non-shared environment played an overwhelming role in both determining suicidal ideation and suicide attempt. Population variance in suicidal behavior, both ideation and attempt, appears to be overwhelmingly determined by variance in the non-shared environment. And again, I'd leave it to you to speculate on exactly what those non-shared environmental factors might be. Returning to Lowland's twin study of the Big Five personality traits, remember that the basic finding was that for each of the Big Five traits, the MZ correlations were about twice the size of the DZ correlations. The typical MZ correlation was in the high 40s, the typical DZ correlation was in the mid-20s. So now let's apply our formulas and try to disentangle the contributions of genetics, the shared environment, and the non-shared environment to individual differences in personality. Let's first look closely at just one Big Five trait, extroversion. We double the difference between MZ and DZ twins and get an estimate that 48% of the variance is attributable to genetic factors. 1 minus the MZ correlation gives an estimate of 52% of population variance attributable to the non-shared environment. And the remainder, well, there isn't any remainder. Apparently none of the variance in extroversion for this group was attributable to the shared environment. And that turns out to be pretty much the case for each of the Big Five personality traits. In each case, we see substantial genetic contributions to population variance, about 40% or so. A substantial non-shared environmental contribution to variance, about 50% or so, but only a very small contribution of the shared environment, typically 10% or less. Genes make a contribution to individual differences in personality, but genes are not decisive. Nor is the family environment decisive for adult personality. The non-shared environment is far more important than the shared environment, and is at least as important, probably more important, than the contribution of the genes. This is the typical finding obtained whenever somebody does a twin study of personality. Behavior genetics studies don't have to be confined to things like intelligence and personality traits. We can also use them to study the origins of attitudes. Here's an example from the Virginia 30K twin study, a study of almost 30,000 twins who lived in the state of Virginia. As part of this study, these individuals were administered an attitude inventory, which asked them whether they endorsed various positions on various socio-political issues: school prayer, property taxes, busing to relieve racial segregation, abortion rights, and so on. There were 25 of these stated in a more or less liberal way, 25 more stated in a more or less conservative way. 28 of the items were expressly political in nature. That is, they had to do with proposals for new laws. And the subjects were simply asked to indicate whether they agreed or disagreed or were uncertain about each of these statements. Here are the correlations obtained in that study. The survey items were collected into two somewhat different scales measuring liberal versus conservative attitudes. Remember, that's the basic dimension of attitudes, liberalism versus conservatism. Then there was the scale of opinionation, basically the number of items in which the subjects actually had an opinion as opposed to being uncertain. And finally, the subjects were asked about their political party affiliation, Democrat, Republican, Independent, whatever. You can see that in each case, the correlations for monozygotic twins are higher than the correlations for dizygotic twins. And here's how the estimates of the components of variance in political attitudes turned out. For both measures of liberalism-conservatism, there was a significant genetic component amounting to 30% or 40% of the variance. And there was also a genetic contribution to the measure of opinionation, or strength of attitude. But once again, the contribution of the non-shared environment was much greater than the contribution of the shared environment. Things were somewhat different for party affiliation. Here there was no genetic contribution to speak of. Almost all of the action is in the environment, and the contributions are about evenly divided between the shared environment and the non-shared environment. It's very interesting that there is any genetic contribution at all to liberal versus conservative attitudes, but it's not surprising that there's no genetic contribution to party affiliation. After all, party affiliation is something that can change very radically with changes in historical circumstances. Between the time of the Civil War and the 1950s, the dominant political party in Virginia, part of the Old South, was Democratic. Beginning in the 1950s with the rise of the Civil Rights Movement, and especially with President Nixon's Southern strategy, the party affiliation of Southerners quickly moved from predominantly Democratic to predominantly Republican. That's probably why the shared environment is as important as it is, in the case of party affiliation, compared to liberalism, conservatism, or opinionation. But even with party affiliation, the role of the non-shared environment is substantial. There may be a strong tendency for children in the same family to all have the same political party affiliation, perhaps adopting the political affiliation of their parents. But something can happen to lead one sibling or a couple to switch, and that's the impact of the non-shared environment. But here again, the outline of the results is generally the same as what we saw for personality traits. Aside from party affiliation, there is a substantial genetic contribution to political attitudes, but it's not decisive. The environment is important. And in the final analysis, as important as the shared environment is to party affiliation, the non-shared environment is a more important contributor to variance in overall liberalism and conservatism. This point is really important, so I want to repeat it. Twin studies reveal genetic influences on personality, including attitudes, and these are interesting. But by far the most surprising finding of behavior genetics research is the evident power of the environment, and especially the non-shared environment. Genes are important, but the non-shared environment is just as important. This typical finding of behavior genetics studies, that the contribution of the non-shared environment is more important than that of the shared environment, is sometimes misinterpreted as meaning that parents have no influences on their children, but it doesn't really mean that at all. What it means, first, is that parents don't have the same effects on each of their children. Presumably, there are some ways in which parents treat all their children alike. And whatever that is, in terms of such things as age of weaning or age and severity of toilet training, whether the children have play groups that they participate in or go to nursery school before they enroll in kindergarten. These things just aren't strongly determinative of personality. What's more important, as we'll see in the next lecture, is that parents just don't have the same effects on each of their children. Parents can have tremendous influence on their kids, but they don't have the same influence on each and every one of them. In addition, it means that there are other forces at work besides the parents. These become increasingly important as the child begins to move beyond the family, for example, by going to school or joining a soccer league. In fact, Judith Rich Harris, a developmental psychologist, has proposed a theory of group socialization, which argues that peer groups and peer cultures, not parents, are the most powerful social forces impinging on a child. In fact, Judith Rich Harris, a developmental psychologist, has proposed a theory of group socialization, which argues that peer groups and peer cultures, not parents, are the most powerful social forces impinging on a child. In fact, Harris argues that socialization is context-specific, and that children may behave quite differently, follow quite different sets of rules, depending on whether they're at home or away, and depending on which particular extrafamilial context they're in. As an example, she points to what's known as code switching among bilingual and bicultural children. Children born to Spanish-speaking parents, for example, may continue to speak Spanish inside the home when they're with their parents, even though they'll speak English with their peers outside the home. Harris's point is simply that something like code switching occurs beyond the domain of language and may affect all aspects of the child's personality and behavior. A child may be one person at home with his or her parents and may be quite another person outside the home when he or she is with friends. For example, minority children from middle-class families may get a lot of pressure from their parents to take school seriously and achieve academically, but they may succumb to pressure from their peer groups to downplay academic achievement and other forms of what's sometimes called acting white. Peer groups and peer cultures are very important, and as the child begins to spend more and more time outside the home, they become ever more important. As a somewhat silly, I suppose, but still illuminating example, Harris cites food preferences. She asks, if parents are such powerful socialization agents, why do they find it so hard to get their children to eat what they want them to eat? This is a real problem that every parent has encountered. And Harris's answer is that children want to eat what their friends like, not what their parents like. In eating, as in language, as in behavior, there's a lot of code switching that goes on. And the more the child is subject to group socialization outside the home, the less influence the shared home environment is going to have. A study of academic motivation in 4th and 5th grade children by Thomas Kinderman makes this point nicely. Kinderman found that students in the school he studied tended to group themselves into cliques such as the brains and the slackers. But he also found that membership in these groups tended to be somewhat unstable, with individual children moving back and forth from one group to another. And interestingly, Kinderman discovered that children's attitudes towards school changed as they changed cliques. Brains lost interest in school if they moved to the slacker clique, and the slackers gained interest if they moved to the brains clique. Why kids would move from one clique to another isn't completely clear, although we know that happens. Just think about your own childhood. But the fact of the matter is, the movement from one clique to another preceded the attitude change. It wasn't caused by the attitude change. It wasn't as if a kid in the brains decided she wasn't interested in school anymore and she was going to go join the slackers. So movement from one group of kids to another changed the child's attitudes, despite the fact that these kids' IQs remained constant, obviously, and their parental influences did so as well, presumably. It was the peer group that caused the changes to occur. Returning to the behavior genetics research, that's the theme for this lecture, we can see the influence of the parents and the peer group on various aspects of adolescent behavior in a study from David Rowe. In one analysis, Rowe found that parents who smoke cigarettes tend to have children who smoke cigarettes too. And his behavior genetic analysis indicated that this influence was mediated by heredity, that is, the genetic component of variance, but not by the shared family environment. So parents did have an influence on their children, but that influence was transmitted genetically, through genetic inheritance, not through the family environment that the parents created for their children. On the other hand, adolescents who smoke cigarettes tend to have peers who smoke cigarettes as well. This is an effect of the non-shared environment, an effect of the environment outside the family. Rowe and his colleagues found similar causal patterns for alcohol consumption, juvenile delinquency, sexual behavior, and pregnancy. In each case, the shared family environment had relatively little impact on behavior. Genetic transmission from parent to child was important, but so was the non-shared environment. Peer groups had a powerful effect on whether adolescents experimented with tobacco, alcohol, sex, and generally misbehaved. So one way in which the non-shared environment shapes personality, attitudes, and general social behavior is through the environment outside the family. Schoolmates, playmates, teachers, ministers, whatever. But interestingly, the non-shared environment also exists inside the household. How that can be, that's the subject of our next lecture.